So, good afternoon, everyone. So, thank you, Zeni, for this very nice introduction. And maybe I can go a little bit more in detail into this project, which is entitled CO2 concentration mechanism in photosynthetic microorganism. And this is algae. So the underlying question is, how do aquatic photoautotrophic microorganisms adapt to varying dissolved inorganic carbon availability, also the CO2 to oxygen ratio, whilst maintaining such an efficiency in CO2 fixation. So the context, as Zeni introduced, is the oceanic carbon pump, but this scheme can also be used for soil carbon pump. Photoautotrophic organism, the primary producer of or primary producer, and they have to adapt to a varying nutrient, of course, scarcity of oligonutrient, nitrogen sources, but also to varying CO2 to oxygen ratio. And this is peculiar for aquatic organism, but also in the soil. And microorganism, unicellular organism, have to do it everything in one cell, so they cannot use cell differentiation like higher plant would do. And so they've adapted different mechanism that is very variable, and so that's why there is such a variety of microalgae and cyanobacteria, which are bacteria. So in this project, we work on the scale of the cell and the enzyme, the molecules, and this can be integrated in probably the other scale that you are familiar with, that you use for models. So typically you model photoautotrophic based on simplified models for carbon, nitrogen, phosphate assimilation, but we work in a single scale. So this is to put you in a context of what we are doing. We have model organism, cyanobacteria, Chlamydomonas renati green algae, and this is a soil algae, and a diatom, here Phaeodactylum tricornutum. So these cyanobacteria and diatoms are representative of the oceanic microalgae. And together, there is estimate that they can fix 40% of the primary producer for 40% of the carbon, earth carbon. And Chlamydomonas renati is a model of a soil algae. That is also a contributor, so there is a study in Toulouse that suggested that there's responsible for almost 6% of the carbon sequestration, so this is a smaller contribution, but still I say no, so maybe. We have these oxygenic cultures here in Marseille, and we use this model to study the physiology of this model. And we also are interested in the molecular event that underline carbon sequestration, which is photosynthesis and photorespiration. So this enzyme, Rubisco, that has been studied extensively in isolation. It's not distributed in the cell, and we are very much looking into how this enzyme functions within the cell. So they're present in a specific compartment of the cell, which is called pyrenoid in green in eukaryotic algae, or in carboxysm in prokaryotic photosynthetic organism. And these compartments have been deciphered very recently, and they are a liquid-liquid phase separation. So Rubisco is not in water, it's in a liquid that is a very high density of protein. This is mediated by disordered proteins, which are depicted in orange here. And this localization confers to this compartment specific physical chemical properties. And we argue that these properties contribute to the efficiency of the carboxylation and photosynthesis, and also maybe can regulate also photorespiration. And it's been shown that this compartment contribute to the flux of CO2 to this. So we are interested in the molecular properties of this compartment and how this can explain such a high efficiency of CO2 fixation. So the objective of the CO2 CM<hesitation> project is to describe the molecular key feature that drives Rubisco condensation. Is Rubisco always in this compartment or not? What are the properties of this compartment, and what is the consequence of this compartmentalization to the flux of Rubisco substrate and product, and also the downstream metabolic fluxes and the outcome of the carbon production. What are the consequences of Rubisco location to the carboxylation or oxygenation, the metabolic flux, and the carbon flux, as I just said. I'm going to the wrong direction. So the approach we want to use is to reproduce in vitro the compartment that I've just described, the Rubisco and the scaffold protein, from these three models organisms. So we'll have in vitro reconstitution of mini photosynthetic Rubisco condensate. We will look at their physico-chemical properties using nuclear magnetic resonance and other biophysical tools. And we will look at the Rubisco activity using membrane inlet mass spectrometry and other tools. So we'll have the physicochemistry and activity. Oh, I was using the wrong. We will relate this to in vivo, so the cellular scale, so we will also work on the cellular scale to monitor the condition in which Rubisco is condensed in the pyrenoid or not. And to do this, we will use time-lapse microscopic images to follow the condensation and diffusion of Rubisco, and we will relate this observation to the metabolism of the cell. And we will do this by varying the CO2 in the lab, because that's the chance that we can have lab perfectly controlled condition, varying the CO2 condition, the oxygen condition, and also the presence of other nutrients that has been shown to modulate the downstream metabolism, production of lipids or other. I also think that this is a part where we can relate to the other project of the FairCarboN, where you have all these ranges of concentration in estuaries or other conditions or in the soil. And to complete the scientific strategy, we also want to decipher the Rubisco activity compared to the Rubisco location. And to do this, we will both do on the in vitro scale or in the cellular scale, we will use camera where the Rubisco activities will be either from a given organism, so this is the column here, will be monitored when the condensation is mediated by the disorder protein from the other organism. And by this way, we will look at the contribution of this specular condensation on the Rubisco activity. And this from, as I said, a cellular scale and molecular scale. So the expected outcome of this project is also we hope to provide adequate value for the photosynthesis photorespiration ratio, and also the global photoautotrophic with the metabolic impact on the presence of the adaptation of the algae to a different CO2 to oxygen ratio. There is one aspect that Zeni mentioned is our contribution to perspective for carbon sequestration. So what we would like to provide is to have a rationale for design of biomimetics, maybe a completely biological synthetic biology approach with no cell, but this biocondensate is very intriguing and a way to pursue for carbon fixation, and also a rationale for the choice of alga strain that is maybe on the fashionable for carbon sequestration. So here probably the choice of alga strain is a question on which we probably have some contribution. Thank you.
Now, It's my fault. You actually have 4 more minutes left.
Yeah,
if anybody
actually,
wants to...
I was, maybe this is important. I forgot to present the consortium. Yeah, this is actually very important. So Gilles is also here from the BIAM Institute. So as the name of our lab suggests, that we are very much on the molecular and cellular scale. So we're working on the bioénergétique and ingénierie des protéines lab. Gilles and Yongwa from the Institut de Biosciences et Biotechnologie d'Aix-Marseille. Mathieu and Amel are from the Laboratoire de Chimie Bacterienne. So, and Olivier Bornet, Institut de Microbiologie de la Méditerranée. So we are on the cellular and molecular scale. Maybe this is the
Great. Now, if you want, the questions are included in the 15 minutes. So if you still have 4 minutes left, if anybody wants to ask any questions, was that all clear? You can still talk for 4 minutes if you want. If you want to say 4 minutes, would you? Have you
I
solved the project?
don't want to bore people.
No, Would you like? Yes, absolutely.
So, what are the implications for the carbon and what you can do to improve the carbon balance, and what is the application of your research? It's
okay.
Did you want to ask a question too?
But my question is a bit different. I was wondering if it would have had any meaning to include the chloroplast from a terrestrial plant in the comparison. With
Chloroplast of higher plants.
Yeah.
Yeah. So actually, maybe the answer for the second question is it's easier for me. So this Rubisco condensation, this doesn't exist in higher plant. But it has been introduced by a lab in Scotland as a way to have an extra layer of carbon CO2 concentration mechanism in higher plants. So they have other metabolic way to concentrate carbon around Rubisco, and so far. The proof of principle works. So yes, they have been able to concentrate the Rubisco, to condense Rubisco, and they know they still have missing elements to this, and they are working on it. So now they have added carbonic anhydrase to it. So they know what they have to do for it to work. Maybe this is the way. I think a lab also in Germany have measured increased potato weight after adding this condensation. So yeah, this is an element. And for our contribution, so we are from we are all CNRS. So maybe I don't want to. So we are all fundamental science. So, and this was also, well, this is a question that always comes to people we talk to. From on the very short term, I don't think I'll have a solution to propose to you. Oh, let's use this algae on your garden, and this will compensate your driving, for example. Where I drive. Nonetheless, I think so. Increasing knowledge is what I would prefer as an answer. But as I said, this we will design new algae strain. We want to design this strain to decipher the location and the activity. And I don't want to say this is one target, but one of the aspects is one of these strains will be more efficient in carbon sequestration than the other, because that's the nature. So that can be one outcome. And also the other outcome is that now there is, so there's a paper from last week where they used synthetic biology, so cell-free system of carbon, so cell-free, just proteins, to make condensate that can sequester carbon and produce starch. So this is something that works. This is something you need a protein to do this, but this is an outcome that works. this is a very much biotechnological approach.
Perfect.
So, yeah, so this is maybe
exactly 5 minutes. We're going to 15 minutes are going to be very fair, but you presented that brilliantly. Thank you very much indeed. Big round of applause for our first. Thanks.